So can you hear me? Okay, good. I can hear me. So today's well, first of all, welcome to the Faculty Research Forum. My name is Eva Maria Suarez, and I'm the chair of the committee. Today we're happy to present Dr. Jacquelyn Cole. For those of you who don't know, although I assume most everybody does, she's an assistant professor of chemistry here at Shepherd. She was a postdoc at the NIH in Bethesda, as well as the New York State Department of Health in Albany. And she's attended the Rensselaer, am I saying that right? Rensselaer? Polytechnic Institute for Graduate Studies. She focuses on bioanalytical chemistry using a mass spectrometry, including studying proteins, hormones, and aptamers. And today she talks about caffeine and wintergreen, small molecule analysis.
All right, hi and welcome. So today I'm going to be talking about caffeine and wintergreen, two different projects that I have been working with my undergraduate research students. So first, to talk a little bit about different projects that led me to this point. So I started out the beginnings of my life in a small city in Northeast Ohio, good times, and from there I wanted to go out and see the world, and so I went to the other panhandle of West Virginia at Bethany College in West Virginia, and Bethany College was a great experience. I started out as a biology major, and I wanted to do genetics. I wanted to learn all of the good things about genetics, and then I saw the light somewhere in organic chemistry and became a chemistry major instead. So I ended up majoring in chemistry with a forensic science concentration and a math and physics minors. And from there, during that time, I was able to seek outside research opportunities through the NSF REU program. So the first summer, I was able to work with Dr. Smiley doing biochemistry, and I really fell in love with biochemistry. I had never actually had biochemistry before I was doing biochemistry research, and I really enjoyed that entire summer. And the next summer, I did a polymer chemistry, which I learned I did not care for polymer chemistry very well during that summer. And so during my senior year at Bethany College, we also had to do, we didn't call it a capstone, we called it a senior research project. And so I developed a process for doing amino acid analysis. So I was working with a lot of mass spectrometry already, a lot of proteins already, and so that's kind of where a lot of my different projects went from there. So I had a MALDI MS project while I was at RPI. I was working with proteins, biological samples, so you see that kind of trend of proteins, biological samples, and mass spectrometry. So I sought out a postdoc that was using the other side. So MALDI is a really great instrumental technique, but it works for large molecules, and small molecules are a lot more interesting to me. And so I was doing LCMS of steroids and metabolites at the New York State Department of Health, which was a lot of fun. And I continued that similar sort of process, but instead using peptides. So taking whole proteins and breaking them down into peptides. And through all of this, I learned a lot about mass spectrometry, about a process called MRM, which we'll talk about in here, as well as with internal standards and the different types of internal standards. So a lot of that is what goes into primarily the caffeine project. So the two projects that I work with students, I've actually had apparently an equal number in both of them when I was going through my acknowledgements page. So we have two projects I'm going to talk about, caffeine and wintergreen. So they seem kind of separate, and in a way they are kind of separate. But we're going to talk about quantitative analysis of caffeine in specifically the Potomac River samples, and this is using an LC-MS-MS, the same thing that I used for peptide analysis and steroid analysis. And then we're also going to talk about wintergreen, where we're doing a qualitative analysis specifically of wintergreen oil with hopes to expand that onto other types of essential oils. All right, so caffeine, we all know and love caffeine. This is a stimulant. It's found in our favorite drink, coffee, possibly tea, as well as soda, energy drinks, supplements, and chocolate, pretty much everything that keeps college students going. So we know that there is plenty of caffeine found in Shepherdstown. It is a purine, so it is related to DNA and RNA bases. It looks very similar to that. It has that heterocyclic amine structure going for it. In other words, we have all of those ends, and they are in a circle connected together. And it also classifies as an alkaloid, which is a type of plant stimulant usually. A lot of our medications were initially alkaloids and then developed to try to be less addictive. So alkaloids have a tendency to be very addictive. And caffeine specifically has a couple metabolites, paraxanthine, theobromine, and theophylline are the primary ones. And then there's some good data down here if you ever want to know any of the fundamental characteristics of it. But one of the key things with caffeine is that it is not wonderfully soluble, even though we do drink it in coffee. At those concentrations, it's soluble, but at higher concentrations, it does start to lose a lot of that solubility. So that has been tricky. You can see solubility at 25 degrees Celsius, so room temperature, is only 16 milligrams, but as you get into that boiling water, you get up to the 600 milligrams. So that's really that difference between coffee and other substances. So this stemmed from looking at caffeine in waterways that have already been studied. So you can see that we have a lot of different places that have already been studied where they've already found caffeine. Obviously, we're not going to present usually data where things aren't being found, so who's to say that they haven't tried and checked other places without finding it. And so if we put Shepherdstown on that nice little map, we get this red dot over there in the United States. So we're kind of going to see, let's round out things, and why not Shepherdstown? It's a perfectly good place to study caffeine. So if you look at the different concentrations that have been found at all of those different locations, in Oregon, it ranges in the very low values. That is going to be in the parts per trillion, whereas in Greece we have parts per billion. So these are environmentally they're probably significant, but from a human standpoint, we're talking about nanograms per liter. This is well below coffee's limit if you were thinking about it in terms of coffee. But you can see that we've had a variety of different caffeine concentrations that were found. A lot of these are probably limited to the method, the extraction procedure, all the different process that they went through, how much they had to concentrate the water. So for example, the one in Oregon, I would imagine that they had to concentrate that water significantly just to see that very trace amount. Greece, on the other hand, probably did not have to concentrate it too heavily to be able to see that. You see there's two primary methods that are used there. There's GCMS and HPLC, or in other words, LCMS. And so those are going to be the methods that we're going to talk about in terms of using. We personally use LCMS for our caffeine quantitation. So thinking about parts per trillion, just to give you one of those kind of scales. So 10 parts per billion, starting out there, that's the maximum contamination level of arsenic in water samples. So that's what the government considers to be safe for arsenic samples, or the upper level of safety. Not really safe, but so that's 10 parts per billion, and a lot of your environmental chemicals are going to be on that parts per billion scale. So if you think of that in real world terms, that's equivalent to 10 seconds in about 31.7 years of your life. So imagine what happens over the course of 10 seconds, and then think about the next 32 years. That's the equivalent of that 10 parts per billion. So we really are talking about trace samples. So in Greece, they would have found 3.07 parts per billion caffeine based on their sample numbers. And so, that would be on the scale of those parts per billion. Meanwhile, parts per trillion is 1,000-fold smaller. So in chemistry, we like to do a lot of 1 to 1,000 with our prefixes. And so parts per trillion is 1,000 times smaller. So this is equivalent to about 31 seconds every million years. So when we're talking about 10 parts per trillion caffeine in Oregon, we are really talking about trace amounts being found. So how did that caffeine get there? Well, we are pretty much primarily to blame. Unless you're in an area where crops that contain caffeine like tea and coffee are naturally grown, any other caffeine is put there from human sources. So the average daily human consumption of caffeine is about 140 to 210 milligrams per day. You might be thinking that's a lot, but that's really equivalent to a cup of coffee. That's a cup of coffee. That's about 6 ounces of coffee, not the jumbo size from Starbucks or anything like that. About 6% of our population ingests greater than 500 milligrams per day, so that's definitely going to be the jumbo size cup of that. Most caffeine is metabolized, but 1 to 10% does remain intact. In other words, as it goes in your mouth, it's going to come back out as is. And so it's a very good indicator of the efficiency of wastewater treatment and pollution in that area, as well as just general disposal of caffeinated drinks. You purchased a coffee first thing in the morning, you forgot about it, it's now cold, you don't want it anymore, and you pour it down the drain or in the grass or something like that. That's also going to end up eventually in that wastewater treatment system. So why the Potomac? Well, because it's right there, right? What better purpose of studying something than something that is literally steps from our door? So the Potomac is a great source for studying, and it supplies the water for numerous towns, as well as all of the plants and wildlife in the area. So the question that we were looking for is, are we drinking caffeinated tap water? So if you've ever seen any of my students, they would be going down to the river. Now, the initial students, they don't like to really get into the water, but my last student, he owned his own pair of water waders, so he was right there in the middle of the river collecting samples in the best way possible. So the best way is that you would take, we have these very large mason jars, you take it down to the river, you unscrew the cap, you put it under the water, rinse it out with the river water several times, then you pour everything out, seal up the cap, not really seal up, but close it back up. You put the whole thing as far under the water as you can, so really ideally you're going to get as far into the river as you can. He was a very tall student, so he got very far into the river. I had to keep warning him not to get swept away by any of the water on some of those days that we had. I'd have to tell him every morning, like, no, the river's moving too quickly, you're not allowed down there today. And then you take the entire mason jar, you plunge it all the way under the water as much as possible, and then you open up the cap. That way it allows the water to go in there and you're not skimming from the surface, you're not getting the lower portion. You seal it up underwater and then you carry it back up to the lab. Now the catch is that the instrument's not capable of analyzing water as is. We can't just take a water sample and put it right into the instrument. First and foremost, there's a lot of other stuff in there. So we have found anything from sediment to pond scum and probably everything in between. So we can't just put it straight in there or it would clog up the column. So we have to be very careful in that sense. So our first step has become just putting it through a simple coffee filter. That does actually pull out most of the sediment, and it actually gets rid of a lot of the pond scum that we were noticing that was occurring as a result of some of the organic solvents later on. So that's the first step. The second problem is that it's not concentrated enough for our instrument, or rather our instrument is not necessarily good enough for that. If we had one of those nice $1 million instruments, we might be able to use it right as is, but for ours, we do have to concentrate it down. So this usually results in taking about 1 liter of water, and we end up evaporating it down to about 50 milliliters. The catch with this is that can take up to 8 hours of just slowly boiling this away. So it is a lengthy process, and we're investigating other options such as using some small chromatography columns and everything to capture the caffeine. It would be a slightly faster process. It would not use heat, but it's only slightly faster, but at least the students can, in that particular case, they could set it up and go to class and come back, and it would probably still be slowly going through. And the next step is to extract it. So we use organic solvents to extract our samples. The extraction process is to leave behind any of the stuff that we don't want to keep in there. We want to see the caffeine and we don't want to keep any of the other stuff, preferably. Now we know some of the stuff is going to come through, and we deal with that on the mass spec end, and I'll talk about that when we get to that slide. But for this, we've used 2 different solvents. We've used IPA, which is isopropyl alcohol. One of the challenges with IPA is that we have to salt it, so we have to put a large amount of salts in it to actually make the chemistry work and actually create the 2 different layers that we need. However, you can imagine if you put a lot of salt into a system, there's going to be that much salt coming back out of the system, and the salt can build up on the mass spec whenever we get to that stage. So the preferred method would be a method using DCM or dichloromethane. This is a pretty standard organic solvent. Both of these have a high affinity for caffeine, so caffeine works very nicely with them. But initially when we were working with DCM, we were getting that pond scum situation, but the coffee filters seem to remove most of those. I'm guessing they end up getting caught in the fibers. And so DCM has become, again, our preferred method, which is nice because then we can extract it in a small amount of DCM. It's a very classic chemistry experiment, and then we can evaporate the DCM away, and then we reconstitute it in a known amount of just a little bit of methanol in water, and we run that directly into the LCMS. So whenever you're looking at mass specs, on that first page you saw that I had a lot of different mass spec projects, and they all actually use different mass specs. So one project was using GCMS, one project was using MALDI-MS, one project was using LCMS, and all of these are actually unique. Mass specs, you can kind of think of them as a semi-system where you can plug in different components and change it. So so much so that actually a lot of research groups, that's what they do, is they just start throwing random things on the front end. In place of an LCMS, they might put on a capillary electrophoresis system, and this is actually yielding a lot of very interesting stuff in the worlds of like proteomics and everything, where you can work with affinities and then put them into this robust mass spec system. So we have some sort of inlet, and we're going to have a source, we're going to have an analyzer, we're going to have a detector, and then it's going to go into the instrument. So in our case, the source is going to be electrospray ionization, and our analyzer is going to be a triple quadrupole mass spec system. The inlet's going to be the LC or the HPLC. So we have the liquid chromatography portion. So the analyte is going to get converted into ions in that source. The ions are going to be analyzed using their mass to charge ratio in the analyzer. Now with large molecules like MALDI, you can actually have multiple charges added onto this. So you get these very beautiful, they almost look like a stegosaurus. When you work with polymers, you get these sort of spikes that just keep continuing on and on. And so that one does get a little bit tricky because you have to take into account how many charges could be bound to a single protein, whereas generally with the small molecules, you're going to be talking about a single charge. So that makes it a little bit easier. Usually what you see is what you see. So it's a little bit more helpful with LCMS. However, the catch is that with LCMS, we can't just put a protein in and hope that it actually is going to work. So we do have to break those down. So we do have to have small molecules, smaller molecules. So later on, the results are going to be using gas chromatography, but I figured I would just put that in this section and then not have to talk too much about instrumentation in the essential oils section. So with gas chromatography, you have this extensive metal coil system, and usually there's going to be some type of stationary phase that is attached inside of that, and you can actually purchase different columns that will modify the different chemistries inside to be able to collect what you would like to actually collect. And that's what ends up working for the gas chromatography. Meanwhile, for liquid chromatography, for like this caffeine project, we work with this tiny little metal tube, and it has the different chemicals packed inside of it. So what the goal of those chemicals are, and again, you can vary those depending on what exactly you're looking to do, but what ends up happening with that is it separates them out, and it's going to separate them out by their affinity for all of these different ones. So we just use a standard C18. So C18 is a carbon, but it's 18 carbons, so it kind of looks like a finger. So you can imagine a molecule trying to get through all of these fingers. And so it's going to get caught depending on the affinity for those fingers, and then we change the mobile phase, we change the liquid that's going in order to flush that out. So ideally, we would only be flushing out one compound at a time. It's going to go into that mass spec, and it's going to be analyzed. And we can extend the runtime in order to try to isolate just one single compound, or we can shorten it and try to run a lot of different things very quickly through it. So this is your standard, just a general GCMS system. So this is what's going to be used with the essential oils. But if you notice from that earlier slide, a lot of the groups that were studying caffeine were also using GCMS. So it is an option for GCMS, but LCMS to me would be a preferred system. It's a more gentle process. So one of the challenges with GCMS is it's a very hard ionization process. It's designed to fragment things, and you use those fragments to kind of back calculate what your molecule is supposed to be and what was fragmented off of it. So you can actually figure out very specifically, to a degree, where things are actually located using GCMS. And so that is very useful, but being a hard ionization process, it does result in many, many ions being produced. Whereas with LCMS, it is a softer ionization process. So since it's really just trying to ionize, it's just trying to get some charge on that initial molecule, it's able to pass through fairly unchanged, and you sometimes will get some fragmentation, but you can generally get through the exact molecule. And we use this to our advantage in our particular one, because if you remember, I said it is a triple quadrupole. So triple quadrupole is this good stuff down here. Ooh, not that button. Wrong button. All right. This stuff down here. And so this is quadrupole 1, 2, and 3. In the first quadrupole, we tell it exactly what to look for. So we take caffeine's molecular weight and we say, look for just that, throw everything else out. In the second quadrupole here, it then fragments that caffeine, and in the third quadrupole, it then tells us what is all there, and we can actually have it selectively look for another specific one. In the world of peptide analysis, which is part of the reason for having the LCMS here on campus, is to eventually do peptide analysis. In the world of peptide analysis, this is the gold standard to find a peptide. If you can say, I'm looking for this fragment, go into this fragment, and I can do that with 3 different fragments on that particular peptide. In the bioanalytical world, that is a positive, definite what it is. And so it's very useful. So if I were to do, let's say, a protein analysis, I would be able to definitely say, oh, this is definitely this particular protein using the different fragmentation patterns. So we use this for caffeine to our benefit, because if we're only looking for caffeine, all of that other stuff that may have came through that extraction is just going to get thrown to the side, not even analyzed, and we don't have to worry about that contaminating our samples. So this is our instrument. This was acquired through the surplus program at US AMRID, as well as the West Virginia EPSCoR grant program and the chemistry department. So it doesn't look like much, but this is the liquid chromatography, and this is the mass spec. And the mass spec is actually about this large. It just goes back on the bench. And our column is this little guy right here. Taped to the cover just so that it doesn't dangle around. So the samples are going to go in. This is an auto sampler, so there's actually a little tray that rotates in here. A needle comes down. It takes up exactly the same amount of sample every time, so it's very helpful to have an auto sampler. It reduces the human error side of things. Then it's going to go in through this column here. It's going to be affected by the chromatography and that C18, and then it goes into here. This is our source where the ions are formed, and you can't see it here, but there's a little nozzle where the ions are formed, and then a gas blows it into the mass spec, and then it goes through the different quadrupoles, and it's analyzed, and then we have a computer that collects the data for us. So as I mentioned, that is the multiple reaction monitoring. So I kind of described it on the previous page with the triple quadrupole. So if I was using our triple quadrupole as just a regular LCMS, our sample here would just go straight through. Nothing would really happen. There could be some fragmentation, but it's going to kind of make a mess. And then we're going to detect every single fragment, and we get this very complex spectrum. Meanwhile, if my sample comes through and I throw out everything but what I'm looking for, when it breaks up just what I want, I'm only going to get specific things that I'm looking for. And so I result in this very clean, very easy to read mass spec. So this is one of the spectra that was collected by one of my students. So this shows theobromine, theophylline, and caffeine on the same spectrum. So she had put all 3 of these into the same sample. And so as they ran through the instrument, this is the order that they came out in. We don't have a lot of control over that order. We could potentially modify some of the chemistry to try to control it, but it's easier in this case. What we would do is we would just send a sample of pure theobromine through, see where it comes out, a sample of theophylline, see where it comes out, a sample of caffeine, see where it comes out, and then put all 3 of them together. Now these 3 were all in the exact same concentration, calibration because we were trying to send them all in the same concentration. You can see that the peaks are not all the same height. That's very characteristic of LCMS. It's along the lines of the whole you get what you get sort of thing with LCMS. So generally you pair LCMS with a good calibration curve, so that way you know exactly what you are looking at and you don't have to do any sort of comparisons, because if you looked at this you might say, oh, she clearly put less theobromine in, but she didn't. She put the same amount of all three of them in. Once you've established a run method, your samples should always come out at the exact same time, so that's also very helpful. And your fragments are also going to show up at the same time. It just shows up in a different menu. You click a button and it brings up all of your different fragments. So when she was working, a lot of her work had to deal with run-to-run sampling to make sure that the instrument was being very consistent, as well as kind of helping out work with the calibration curve. Now the calibration curve has to be ran every single time you run, so you can't run it on a Tuesday and then run the samples on Friday and hope that nothing has changed. So the instrument actually runs best if you're running it continuously. So if you could actually keep samples on it 24 hours a day, it would run better than if you just ran it as needed. So a lot of our goal with this project is to collect up samples and to run them all at once, as opposed to running them every single time that we have them, because we'd have to make a new calibration curve each time. So her standard deviation was actually very good. It was within 7% for her standard deviation. So it really indicated that run-to-run variations were not terrible at all, and that she was putting in the same amount of internal standard each time. So that was very helpful. It really just showed how robust our instrument is. Our instrument is probably about 20 years old, based on the different sources that told me different ages and everything when we got it. So given that it's probably about 20 years old, it's still running like it should. So that's a good thing. And then the next part is the calibration curve. So in this calibration curve, what she did was she varied the amount of caffeine, but she kept the amount of theobromine the same. So the top one has the lowest amount of caffeine, and you can see it's not even showing the caffeine peak, but the theobromine was kept the same each time. So then as the caffeine concentration's increasing, that theobromine peak starts to decrease, and then this one starts to increase. And you can see along here, these numbers also start to increase, and they do increase in a somewhat linear fashion. So that also shows that the instrument is running exactly how it should be. So every student that I train through this project has to go through and make up their own calibration curve and get comfortable with this entire process. So that's probably 95% of the training. The sampling part, I usually just tell them, oh, go extract this, and they remember, oh, I did this in organic, so I can do this. But with the instrument, that is the majority of the work load for teaching the students. So that student did not actually find any caffeine in the Potomac, but when we were going back over her project, we realized that we didn't think we were concentrating it enough. So for that particular concentration, there was nothing present, but we needed to concentrate it more. So the student in 2018, the summer student that summer, actually found our first positive caffeine peak, and he was able to replicate this at least twice. So he definitely believes that this is a caffeine peak that was present from the water samples. So this was very exciting. His calculation is that this amounts to about 2 plus or minus 8 parts per billion in terms of the caffeine. Now, there's a fairly large error bar there, but at the same time, he only was able to see this twice. And if you recall from this last summer, I got that another slide. This last summer was incredibly rainy, so we had a lot of difficulty just finding time to be able to sample and trusting that our samples were actually what we were seeing. So if we took that original caffeine concentration and we put his number onto this scale, it would be located right here. So that's kind of exciting because that's a significant amount. That also indicates that we could be having a problem with wastewater treatment. Now the catch is that it may not be Shepherdstown wastewater, it could be Hagerstown or anything upstream of us. So there's a lot of work that needs to be done, including replicating this result several times preferably. So if you remember, this summer it was definitely one of the wettest on average. It surpassed the average by close to 20 inches of rain, so we're not entirely sure why we saw it this summer, but we didn't see it 2 summers ago. So it could just be the vast amount of rain. It could have been bringing up stuff. It could have also been taxing our wastewater systems. So a lot of the wastewater systems, the water does have to still be processed. The rainwater would still have been processed. So it could have been taxing it, and not everything may have been actually collected and treated. So we definitely need more data to say that we are confident that there is caffeine in the Potomac, but our initial data does suggest there is, so that was very exciting. So future caffeine research, a lot of good projects that we can go with this. I'd like to collect more data and track caffeine concentration over months and years to see if we have any trends. If we had any trends, I would expect to see it highest in the August to December and the January to April months, because that's when we have students here. But that would be dependent on the Shepherdstown wastewater treatment. If we didn't see that, then yay, that means that our wastewater at least is working fairly well. I'd also be interested in increasing the sampling locations both up and downstream of Shepherdstown. To the best of our knowledge, there's no other research being done on this particular waterway, so this could be very exciting. We could also increase this to not just caffeine, but looking at other chemicals like sucralose or steroids in the water samples. Possible interdisciplinary projects, we could work with biology. One of my interests is to see if we grew plants in caffeinated water to see if that caffeine uptake also occurs. So would we expect if somebody did pour a cup of coffee right next to some grass or something like that, is the grass then taking up the caffeine, and then we're passing that on to things like rabbits and mice and everything that could be consuming that grass? And then I'd also be interested in working with ENVS to extract caffeine from soil samples to see just what sort of depth the caffeine goes and how long the caffeine actually stays there, because we know that it should naturally biodegrade, but in soil samples there may not be the organisms present to actually be able to degrade that as quickly as, let's say, water. All right, so that was project number 1. On to project number 2. So this project came about when I was teaching, of all things, college chemistry, and there was a little sample in their textbook that talked about wintergreen oil and how toxic it was. And I was sitting here thinking, can't you just buy that on the internet? Can't you buy that in stores? Isn't that what they use for candy making and everything? And so I started researching the toxicity of wintergreen oil and the information around that, and so we created a project based on essential oils. So wintergreen, we all know wintergreen as the stereotypical lifesaver, right? The nice white lifesaver. It has a sweet odor, so you're probably familiar with wintergreen. It's not quite as spicy as peppermint. It was first isolated in the 1800s. It used in a lot of different chewing gum, mints, mouthwash, food products, as well as these deep heating liniments for your muscles, so muscle rubs and everything. My first true experience with the physics of this was that my teacher way back in, I think it was 6th grade, she took us all into this tiny little closet, gave us one of these, told us to put it in our mouths and crack down on it, and we would see that tribioluminescence that occurs whenever you snap this. And so it has a combination of the sugars and the methyl salicylate that causes that. So it's really interesting that is kind of unique to that wintergreen. It's also toxic to humans due to the salicylates that are present. So that's the structure, and you're going to see the structure a lot over the next slides and everything. So it's not a true mint. So if you look at menthol as a true mint, mint activates a specific ion channel that is a cold response, and it's really interesting to learn about these. So this could be maybe a future biology project as well, as looking at the different ion channels. But mint activates an ion channel that is a cold response, whereas methyl salicylate activates different ion channels, one of which is called burning cold, and one of which is called hot, which is the same response as activated by capsation. So that's why wintergreen gives you that kind of confusing cold yet warm sensation, which is why it's also very preferred for muscle rubs. It really helps out to try to ease the pain there. And there's actually a lot of question as to whether or not it's actually easing things, or if it's tricking your body and it's actually hurting your body, but then your body thinks about that as opposed to the muscle pain, and therefore it's more of a homeopathic remedy and not actually doing anything useful, which would be interesting, because then all of a sudden we would probably pull all those off the market and say, no, you can't use these anymore. Salicylic acid is often used in beauty products and wart removal products and everything. It helps to remove the outer layer of skin. So this was first isolated in the 1800s, but people have been using this for centuries and centuries. They've known about this, but they were just using the bark of the willow tree. They weren't actually isolating it. And this is one of the list of essential medicines. However, salicylic acid is also one of those sources of those salicylates. So if you use products that have salicylic acid in them, as well as muscle rubs, as well as other salicylates, you could accidentally overdose yourself. So one way of synthesizing methyl salicylate is to take that salicylic acid and add methanol. And this is something that we do in the lab with the students during one of their student labs. It's a very simple process. You mix the two of them together, add a little bit of heat, add a little bit of acid, and poof, you've got that beautiful wintergreen smell. So part of our interest in this project was to use all of the different analytical instruments that are available to us to see if these essential oil companies were doing just that. If they were doing something like that, we would expect to see methanol and salicylic as well as the methyl salicylate present. So we were curious to know if that was part of how they were doing these, or if they were in fact naturally extracted as they claim. That salicylic acid can go through a very similar process using acetic anhydride, and this makes that medicine that we all know and love as well, which is aspirin. And so you can see the differences between methyl salicylate and aspirin are which area on the ring that the ester is present on. But otherwise, the process is very simple. We actually make this also in lab with the students. So it's not a far jump to say, could this also be present in our different samples? So that's why it's very helpful to have all of the analytical instruments that we do have available at Shepherd. So aspirin, it even has the word salicylic acid in it. It's acetyl salicylic acid. And it was initially created in the 1800s, and then Bayer took it over right at the turn of the century and named it aspirin, and it's still typically known as aspirin, even though that's technically their trademarked name. It's estimated that probably close to 50 to 120 billion pills are taken each year of this, so that starts to give you an idea of the amount of this that we're also taking in. So it's another one of those ways of accidentally overdosing yourself. My muscles hurt. I'm going to take some aspirin. I'm going to put this liniment on me, and the next thing you know, you have salicylate poisoning. So it's very important to be aware of your total salicylate amount that you're taking in a day's time. So the way that the essential oils are created is usually by steam distillation, or this is what the companies claim at least. So this is a very simple process where you basically just slowly steam the products. The essential oils should move up, and then they can be concentrated further down. We could hypothetically set this up in the lab. This is one of those future projects that I'd like to see done, where we actually have students taking birch bark or willow bark and being able to remove the methyl salicylate directly from there and see if that compares to what we're seeing in the commercial samples. So we have a bunch of different instruments, and I'm not going to go through all of them because there are a lot of them. I'm not going to go through all the different techniques that are used, but one of the methods that we ran methyl salicylate through was UV absorbance. And so UV absorbance, it does have a strong absorbance peaks at 208 and 230. Which is consistent with the literature, so we're hoping to be able to use something like this to quantify our samples later on and be able to say there's this much in each of these samples. The catch is that the UV absorbance, compared to our samples, we know our samples are so concentrated that we have to do like 1 to 1,000 or 1 to 10,000 dilutions just to get this to show up on the instrument, and even this is actually too concentrated for the UV. So it really is a matter of very, very, very concentrated samples that we're working with. And it's difficult to get the students to always understand that that's why we do have to do those dilutions, is just to get it in range. I believe the first student, well, she wanted to just run this straight out, and I said, no, you're not using that many drops of my sample in order to just run this. So I said, do, like a 1 to 100 dilution, because we had no idea what exactly to run it as, and it was just a straight line across. There was nothing to even see. This is definitely a challenge because students are not always the greatest at performing these dilutions that are needed. So we also use the IR spectroscopy. that is available to us. And so what I was focusing on were these 4 oils. So this is acrose. Acrose is not officially an essential oil. That is a chemical. That comes straight from I think it's from Sigma or Fisher, but it is a chemical. So we had no idea what we were going to be finding out of that. We were almost expecting more on the synthesized version of that. We also were looking at doTERRA, Loran oils, and Young Living. So all of these are able to be purchased on Amazon without any problems, various ranges, but in terms of cost, but I think the most any of these costs were about $20 and the least were closer to $5. So for about the same amount for all of those. So we can see that with the IR, there's not much different visually. There may be some differences in the region over here, the very spiky region, but there's nothing that's significant enough that really drew our attention in terms of this. So just taking one of them, I used doTERRA as the sample one, just the example, to show you some of the peaks that can be identified that definitely tell us this is primarily methyl salicylate. And again, there could be other compounds that are influencing some of these, so I don't want to read too heavily into the IR. But we do see an alcohol peak at 3100. We see a carbonyl at 1700. If it was salicylic acid, it would be shifted a little bit, so fairly confident it's not salicylic acid. We see the aromatic group in the 1400 to 1600 range. We see the CO stretch in the 1200 to 1300 range, and again, that would be shifted to the left if it was salicylic acid. And there's no peak at 3500, which would indicate the salicylic acid presence. So if you look at all of those are the peaks that I'm talking about, and so the lack of the salicylic acid is very promising to indicate that this isn't being synthesized. The students wanted to run NMR, so they ran NMR on all of these, and again, we basically got the exact same result for all of these, so just using one of these, looks like this was the Young Living sample. So we see a peak around 3.7, so this is going to be the methyl group, and that would be absent if it was salicylic acid. We see the aromatic ring, and we see the alcohol group, but not much else going on with the NMR, and so you can see the different peaks where those are present at, and so if this peak was absent or lower, then we would be leaning more towards this where there isn't that methyl group here, whereas because it is so strong, we're going to have that methyl group present there. So those are fun instruments, but they're not telling us a lot of information, but the GCMS, on the other hand, that's where these are going to really shine. So when you put all of them in, again, we don't see a lot of differences going on. We have the acrose, the doTERRA, the Loran oils, and the Young Living, and there's not necessarily some significant differences. You can tell that B and D, which are the two from the essential oil companies specifically, they have extra spikes, extra peaks present on those, whereas the other two samples look a lot cleaner. They don't really have a whole lot of extra peaks going on. So that did definitely stand out to us, which we hadn't really seen in the other samples. And so this is just the mass spectra of the methyl salicylate peaks. So we can see there is definitely methyl salicylate in all of them. So what we wanted to do was, first and foremost, these were way too concentrated, and this was actually the students, what they did was they just took the sample, they dipped the needle into the sample, and then they just dipped that into the GCMS. So there was no dilution, but there was also no quantifiable amount being put into the instrument. So we know first and foremost, these have to be diluted, and so that's what one of the students is currently working on. So she's diluting these. She's also extending out the run time to try to draw out as many peaks as possible. So I'm looking forward to her information as she is finishing up that project. The GCMS of the methyl salicylate is identical to the NIST standard that's available on the internet. So we knew this was definitely methyl salicylate. It has the same fragmentations. So we see the fragments at, well, we see the actual compound at 152, and then the fragments at 120, 92, 65, and 53. So we see those, and there's a 39 on the NIST one that's very prominent, and we did not sample as low as 39, so we don't have that particular peak. But we're fairly confident, obviously, this would be methyl salicylate. But the question is, this is using the doTERRA sample as an example. What are all these other peaks that are being shown on either side of the methyl salicylate peak? So when we look at these, we want to really dig down into that. And so one of the things that our instrument can do is it can bring up a library. And so I went through and I looked at the library, the key hits for all of the different samples, just these 4 samples. Now we do have an additional 6 or 7 samples in the lab that we're working on, but these were our like kind of first run sample choices. And so with acros, it is primarily methyl salicylate, and the only other chemical that came up was glycerin, which makes sense. They probably used it either in the extraction process or the purification process, or they added a little bit extra in maybe to get a little bit more for the money and everything. Who's to say? Glycerin's a pretty typical thing to see in there. Now, the percentages that you see next to the methyl salicylate are percentages based on the other things that were also present. So it gives you an idea of approximately where the concentration is, but it's not quantitative. And that's also another thing that the student is going to be working on in terms of quantifying these 4 samples in particular. Now, doTERRA has this beautiful list of things. So we have a couple extra things showing up like pinene, benzoic acid, eugenol, and some other alcohols. So I would expect this out of a sample coming from a plant. I would expect to see other plant-like compounds that would be present. So this actually kind of made me happy because this does really strongly indicate that they are not synthesizing this in the lab. They probably are steam distilling this as they claim. Now, the Loran sample, this one's a little interesting because the only 2 compounds that came up was limonene and methyl salicylate. Not a lot of choices there. Limonene is definitely something I would expect to see from a plant sample, but just methyl salicylate is the other one. I'm not entirely sure what to deduce from this. Maybe it's being synthesized, maybe it's being purified, so maybe it's being starting with a plant and then being purified and they're losing those other compounds. I am not sure, but Loran oils is marketed as a flavoring for foods. So that's quite literally, you purchase it for flavoring. So it's interesting that it doesn't have all of those other plant compounds that I would expect. Now, the really exciting one was Young Living. There's a reason why it was not on the previous slide, because we were getting lots and lots and lots of different compounds. And again, everything that I would expect in a biological sample, so nothing that upsets me. We have pinene, alpha and beta pinene, we have benzaldehyde, we have phenol, we have some other various different compounds that, now again, a lot of these could be only one single compound that was present, but it still does not upset me in any way because it really does kind of show that we're probably talking about a plant-based sample here. We've got some limonene, we've got some eucalyptol, eugenol, and we also have some salicylic acid, but it was such a small amount that it really doesn't indicate that they were using it. It's probably just naturally occurring. So that was really fun. So one of the things that I'm going to focus on, I think, with future projects is maybe using these Young Living samples, because this gave me a much nicer list to work with as compared to, like, let's say the doTERRA sample. So with the methyl salicylate, if you look at the time, they're all about the same, so that's good. That indicates that that's going to be methyl salicylate. So just like the other mass spectrometries, time matters with the chromatography. It should be very consistent. So once you identify a compound, it is likely to come out at the exact same time. And the percentages were all in that upper 80s to 90s%, which is also what I would expect. And like I said, this does have some influence on the other compounds that were present, but it should still be the majority of the sample that I would expect. Other notable compounds present, so pinene, were in the doTERRA and the Young Living samples. This is a naturally occurring insect repellent in plants, so totally normal to be there. Benzoic acid, benzaldehyde, both biological products that you would expect. Eugenol is also present in doTERRA and Young Living samples, so this is a naturally occurring clove-like scent in plants. And then salicylic acid in very trace amounts was found in the Young Living sample, but again, probably a naturally occurring version of that. So to wrap up things, so salicylates. Salicylates are going to be a salt or ester of salicylic acid. So aspirin, bismuth subsalicate, you may be familiar with that being Pepto-Bismol. as well as methyl salicylate and everything. So you can see that these are very similar to each other, and the part that is the salicylate part is going to be kind of specifically this sort of area. It's conserved across all of them. You also have some form of ester or carboxylic acid occurring in all of them. And so this is where we get into a lot of those problems where you can accidentally overdose yourself by consuming too many products that contain these salicylates. So again, if you take aspirin, Pepto-Bismol, put on a muscle rub, drink some wintergreen tea, you're pretty much getting everything from everywhere. And salicylates are not the safest things. So salicylates inhibit the activity of the COX-1 and COX-2. And so this helps to decrease the formation of prostaglandins from the arachidonic acid in our bodies. Other COX-1 and COX-2 inhibitors are ibuprofen and naproxen, so your Motrin or Advil and your Aleve. So you also have to be very careful if you take those and then put one of those muscle rubs on, because in that case you're actually getting the problem of multiple NSAIDs, because your body is going to take that muscle rub and it's going to actually convert it into the salicylate, which is then going to act as this NSAID. So you're actually doubling up on things. We also have to be aware of Reye's syndrome in children. So this is typically considered aspirin, but they've seen this in Pepto-Bismol, and I would imagine in these muscle rubs, especially when you combine that with a viral infection. And so this causes swelling in the liver and the brain, and it can result in permanent injury or death. So again, just being aware of those salicylates, and we can take these both internally and topically, meaning that it's very easy to overdose without actually realizing it. Salicylates occur naturally in plants, and several studies have suggested that humans can metabolize these in actual measurable quantities. So that could be a future direction of a project, is that we could actually see what metabolites are produced after using these types of liniments and everything. And salicylic acid can also be produced through the hydrolysis of aspirin or methyl salicylate, like in a strong acid or a base, like what's found in your intestinal tract. So we can actually ultimately end up making that ourselves by accident. 5 milliliters, that's about a teaspoon of methyl salicylate, is equivalent to about 6 grams of salicylate, or about 20 milligram aspirin tablets. So that's just 1 teaspoon. Now, you have to have some very strong abilities to actually consume that much methyl salicylate, but you might be thinking, well then, why would anybody sell any sort of measurable amount? And I can tell you that this is 15 milliliters, and so that's 3 doses. This is equivalent to 900 milligram or 20, this would be equivalent to 60 tablets of aspirin just in this little bottle, and this just costs $20. There's a safety seal on it, unlike most of their oils, but that's about all you get for that. So toxicity can occur in doses around 150 milligrams per kilogram. So for a 22-pound child, just 1 milliliter of this could kill. So it's very easy for your child to get a hold of something like this. And numerous journal articles have been published studying deaths by overuse of topical wintergreen muscle products or ingestion of the oil. So you do have to be very careful when you're looking at those salicylates. So future research, I'm interested in determining the exact concentration, because it's great to say, well, toxicity-wise, that's about what is in here, but it'd be nicer to be able to say this is exactly X amount of milligrams per milliliter in these different components. And also stems a lot of interdisciplinary projects, like in biology, we could do toxicity studies on cells or bacteria. We could also study the concentration present in actual plants and not just in the samples that we have present. Next research project that I'm looking into is going to be studying the essential oils from grapefruit, lemon, lime, orange, and tangerine using the GCMS, so that we can actually write down an entire list of everything and compare and contrast them and potentially quantify all of those, because I think that makes a very interesting project. This is really great because instead of the students dreading coming to lab, they're kind of excited to work with the essential oils because they smell good, right? They don't have to work with, stinky chemicals. They get to work with good smelling stuff. So we could also include these in things like, again, toxicity studies with biology, or growing and extracting our own citrus essential oils using the other departments. I would not be here without my student researchers, so my wonderful student researchers that have been on the caffeine and the essential oils projects. So we have a lot of good names here listed. Megan and Devin Umstead for caffeine have done the bulk of the work, and then Jamie has done a lot of the essential oils work that you see presented here. A lot of the different IR and GCMS came from her work over the summer. And thank you.
All right, we have about 5 minutes of questions.
Jackie, I had a couple questions about your caffeine. This is louder than I thought, sorry. About the caffeine side of the project. One was that you gave the MCL for arsenic. Is there an MCL or an MCLG for caffeine?
Not that I'm aware of. I can just tell you from antecedently, my uncle drinks like 15 cups of coffee a day, and he's still here at 60. So I'm guessing that probably not. It'd probably be probably more biochemically dependent. I have heard of people who are sensitive to caffeine. So in those individuals, it'd probably be very low, whereas I think normal people can tolerate a fairly high level. So.
And then the other one is when you were talking about filtering the samples in order to get rid of particulate, and then you also kind of came back to that a little bit when you said one of your collaborative projects you envision was looking at soil samples. I was wondering, is there a likelihood that caffeine adsorbs onto particles like sediment, and that when you're filtering it, you might be getting rid of some of your caffeine?
Oh, that's a very good question. Since we were just looking for the presence of caffeine, if we lose some of it, it's not going to be the end of the world. But as we start to get into like soil sample analysis, that could be a really good project where we could go into the river, collect the actual sediment at the bottom, and then be able to pull it back off. Extracting with the dirt and river samples is a little bit more different because you do have a lot of there's going to be a lot more filtration going on. So in that case, we're probably going to lose more things. But since we're really just looking for anything, it's definitely an area that we will explore. So yeah.
Any other questions?
I have a question. I don't need a microphone. I have a lot of questions. And this is just a technical question about the HPLC that you used. With the caffeine study, you used theobromine and theophylline. Were those just sort of as markers so that in cases of low concentrations, you would know where it should show up?
Yeah, so the question was about theobromine and theophylline, if they were used as markers. So part of the initial work was just comparing them to see if they would all come out nice and clean and everything. And then we ended up using theophylline as an internal standard. So we do have another internal standard that's a better internal standard. It's D3 caffeine. So what they did was they took 3 methyl groups on a specific branch of the caffeine, and they substituted them for the deuterium to make it slightly heavy. Now it should come out at the exact same place as caffeine, and so we would end up seeing a double peak at the same place. And so ideally this is going to give us the best sample, because if we can put that in at the beginning, we can do the filtration, and it is basically going to be processed in the same way. The problem is that we had 2 problems. The first one is the first student was starting to see some degradation of the D3 caffeine, and then the second student, when he went to revisit it, it doesn't seem to be very friendly to the LCMS like it should. And he also discovered by doing the NMR, so just our NMR, he discovered that the methyls that the company claimed that they switched out were not actually the ones. They actually switched out a different one, which was kind of entertaining because, I mean, we're just a tiny little institute and we basically found out that they kind of sold us the wrong stuff in the end. But we were using the theophylline because theophylline should not be present in any sort of measurable compounds. And we do run a water blank. So we do run water without any internal standard in there to see if the theophylline or the caffeine are present. And then we put the theophylline into the sample. So, yep.
No? Well, thank you very much.
Thank you.
Thank you.